Okay, today we're going to learn a little bit more about the structure of DNA and how it stores and copies the genetic information. So we're going to jump into this presentation, encouraging you to take notes as you go. So the question we're going to be able to answer at the end of the lecture today is, how does the structure of DNA store and copy its genetic information? So this is a good refresher of some of the terms that we've used so far. So we started out with cell, and inside of a cell, a eukaryotic cell contains a nucleus. Inside of that nucleus, there are chromosomes, and then those chromosomes are made up of DNA. So in your notes, you want to draw or write the word cell, draw an arrow to nucleus, to chromosome, to DNA, and think about it. This is putting these terms in order from largest to smallest. So cell, nucleus, chromosome, DNA. There's 4 scientists that were given credit for discovering the structure of DNA, or deoxyribonucleic acid is the formal name. They're James Watson, Francis Crick, Maurice Wilkins, and Rosalind Franklin. Now, Rosalind Franklin, on the next slide, we're going to see that she is the one who actually is credited with this picture. She used a process called X-ray crystallography, which is very important in cracking the case to figure out what the structure of DNA was. On the left-hand side, you'll see all of these different clues of what was known about the structure, but until they kind of had this picture, it was the missing piece that showed that it looked like a twisted ladder. So here are some things to add to your notes about what DNA structure is. It's a long, thin molecule. DNA contains 4 nitrogen-carrying bases. Sometimes we just say they're the bases, but they have nitrogen in them, so that's why we say nitrogen-carrying bases. Those bases are adenine, guanine, thymine, and cytosine. Oftentimes the capital letter A is used for adenine, G is used for guanine, T is used for thymine, and C is used for cytosine, so you can abbreviate those. Notice they all have this ine, I-N-E ending, and that's an indicator that they are these bases. DNA also contains a phosphate group. DNA contains a 5-carbon sugar called deoxyribose. And another thing that was known was the amount of guanine always equaled the amount of cytosine, and the amount of adenine always equaled the amount of thymine. So nitrogen-carrying bases, let's take a look at them. This adenine and guanine actually belong to a category of bases called purines. You do not need to memorize these structures, but they actually contain 2 rings, whereas cytosine and thymine are pyrimidines. Now, thymine is only found in DNA. That is something to make note of in your notes. It's only found in DNA. And cytosine and thymine are just single ring structures compared to these that are double ring structures. So how does this all work? Well, this was a discovery that when they were trying to figure out the structure of DNA, they found that thymine and adenine actually joined together. And you can see these dotted lines are indicating that there's a bond that forms. It's a hydrogen bond that forms between these 2 bases. So we call these base pairing rules. A always pairs with T, and T always pairs with A. Whenever they're near each other, it's kind of like they stick together, like magnets kind of being attracted to each other, or Velcro. These bonds are weak enough that they can break, but they're strong enough to hold them together. Cytosine and guanine acted the same way. They formed hydrogen bonds in 3 different places. And so we say the base pairing rules are C and G, or G pairs with C. So what this we add into this is that that's where the steps of a ladder come from, but the sides of the DNA molecule are made up of deoxyribose and phosphate, so an alternating sugar and phosphate. So this is the sugar. It's a 5-carbon sugar. So you'll notice it has 5 sides. So 1, 2, 3, 4, 5 carbon sugar. And then phosphate groups. The phosphate group is this P in the center, and then it has oxygens. And notice how they are going to be attached to the sugar, and you have multiple groups. So let's take a look at the next slide. It shows you a little bit more simpler diagram. So you have phosphate, sugar, base, phosphate, sugar, base, phosphate, sugar, base, phosphate, sugar, base. Now on the other side, you're going to notice that you actually have the same thing, except in this case, it's going in the opposite direction. Sort of like our streets are two-way streets. You have traffic going in one direction and traffic going in the other direction. That's how DNA is working as well. So this isn't upside down. This is the way I'm trying to show you that it goes in opposite directions. So in your notes, I want you to write down that DNA is a double helix. That's its shape. And DNA is a nucleotide. Sorry, let me take that back. DNA is a nucleic acid. The nucleic acid's monomer is a nucleotide. The nucleotide is made up of a sugar, a phosphate, and a base. So let me say that again so you make sure you have that in your notes. Okay, DNA is a double helix. It's in the category organic compound of a nucleic acid. And it's made up of smaller subunits. Monomer is a nucleotide. So this is a sugar, a phosphate, and a base. So those 3 things combine to make a nucleotide. You'll notice that the 2 strands of DNA are connected by these hydrogen bonds, and the 2 strands connect between the bases. DNA is it's awesome. It's able to replicate, and it's able to basically conserve half of itself and make a new strand, and take the other half and make another new strand, so you are left with identical strands. So hopefully you remember talking or learning about this in mitosis when we looked at the cell cycle and how there is a phase called the S phase when DNA synthesizes or DNA makes a copy of itself. And this is a little bit more of a detailed look at what happens. There's an enzyme called helicase, and it unzips the original DNA strands. And then there's a molecule called DNA polymerase that comes along and adds new nucleotides, and you can see that happening here with this black strand, adds new nucleic acids to each of the growing strands. So when you're done, you have, from one strand, you have 2 identical strands, and that is how DNA passes on genetic information that's exactly the same because it goes through this process. It's a copying, a replicating, duplicating, whatever you want to call it. It's making an exact copy of itself. There are some additional videos if you want to learn a little bit more about what's called semi-conservative replication. And otherwise, if you have any questions, please reach out to me.